Hi everyone, it's Miss Lopez. This is your Chapter 6, Part 4, Notes on Catabolism of Glucose. In this video, we are going to focus on the final biochemical pathway in cell respiration. We started off with Chapter 6.2 on breaking down carbohydrates, specifically glucose, with the first biochemical pathway, which is glucose, or glycolysis, the second pathway, which is the oxidation of pyruvate, and then the third pathway, which is your tricarboxylic acid cycle, also known as the Krebs cycle. In this video, we're going to finish up 6.2 by looking at what happens with those electron carrier NADH and FADH2. For this part of your notes, we are going to be focusing on the inner mitochondrial membrane and looking at what happens between that inner mitochondrial membrane and the matrix of the mitochondria. Just as a reminder, in the processes of glycolysis, we produced 4 ATP through substrate level phosphorylation, and we reduced 2 molecules of NAD+ down to NADH. In the oxidation of pyruvate, we reduced 2 molecules of NAD+ down to NADH. And in the Krebs cycle, we produced 2 molecules of ATP through substrate level phosphorylation. 6 molecules of NAD+ were reduced to NADH, and 2 molecules of FAD+ were reduced to FADH2. In this last reaction, or in this last biochemical pathway, which is known as oxidative phosphorylation, there is a series of redox electron carriers called the respiratory chain embedded in the inner membrane of the mitochondria. We're going to start by taking a look at what these electron carriers are and where they are and the order in which they are embedded in that mitochondrial membrane. So we have a cross-section of the mitochondria with the cytoplasm out here. This is the matrix of our mitochondria, and then it's separated by this inner mitochondrial membrane, which, remember, would have been the original prokaryotic membrane of the prokaryote that the mitochondria once was. What's going to happen is the molecule of NADH is going to get oxidized down to NAD+, and in doing so donate its electrons to this first transmembrane protein you see in the inner mitochondrial membrane. This is NADH dehydrogenase. And not only is it a transmembrane protein, it's also a proton pump, meaning it's going to pump protons from the matrix of the mitochondria into the inner membrane space of the mitochondria. That electron is going to get carried over to the second transmembrane protein in the system, and this one is that cytochrome C. Cytochrome C is also a pump that will transport protons across the mitochondrial membrane, and then the electron is once again carried to the final electron or transmembrane pump and electron acceptor in this membrane. It's going to pump the hydrogen or the proton across the membrane, and then it is going to donate that electron to oxygen, or terminal electron acceptor, and produce water in the process. FADH2 is going to do the same thing. It's going to get reduced down to FAD+. Your FAD+ and your NAD+ can now go back to glycolysis, to the Krebs cycle, to the oxidation of pyruvate to get to gain electrons again, to get re-reduced in the next molecule of glucose. Meanwhile, we are creating a chemoosmotic gradient with the protons across this membrane. These protons are not going to be able to diffuse back down their concentration gradient because of the hydrophobic layer or the hydrophobic tails of the inner mitochondrial membrane. Therefore, in order to go down their concentration gradient, these protons are going to move through a specialized channel called ATP synthase. ATP synthase is going to use the proton motive force, in other words, the kinetic energy of these protons, to phosphorylate ADP into ATP. This process is known as oxidative phosphorylation. Electron transports. Electrons from the oxidation of NADH and FADH2 pass from one carrier to the next in the chain. The oxidation reactions are exergonic. Energy is released and is used to actively transport proton ions across the membrane. Oxidation is always coupled with reduction, and when that NADH is oxidized to NAD+, ultimately what we're going to see is the reduction of oxygen forming water. In other words, once that final electron acceptor, which is oxygen, accepts the electron and hydrogen, it's going to form water. So the actual terminal electron acceptor in this case is the oxygen, forming the water in the process. ATP synthase uses the hydrogen ion concentration gradient to drive the synthesis of ATP by chemiosmosis. Chemiosmosis is the movement of ions across a semi-permeable barrier from a region of higher concentration to a region of lower concentration. ATP synthase converts the potential energy of the proton gradient into chemical energy in ATP. So this is a close-up picture of the ATP synthase with the hydrogen ion concentration gradient, as you can see, established by that electron transport pump, higher in the inner mitochondrial space than what is inside the matrix. Therefore, when the protons move down their concentration gradient into the matrix, they will cause this large protein known as ATP synthase to spin. And in the spinning process, we're harnessing the movement, the kinetic energy of those protons and using it to phosphorylate ATP or ADP into ATP, thereby converting kinetic energy into chemical potential energy. ATP synthase structure is similar in all organisms. In prokaryotes, the proton gradient is set up across the cell membrane. In eukaryotes, chemiosmosis occurs in the mitochondria and in the chloroplasts. The mechanism of chemiosmosis is similar in almost all forms of life. Chemiosmosis can be determined experimentally. A proton gradient can be introduced artificially in a chloroplast or mitochondria in test tubes, and ATP is still going to be synthesized as long as there's ADP and inorganic phosphate present. So looking at our accounting that we've done so far, what we're trying to say is that for every NADH molecule that is oxidized, you can produce 3 ATP. So let's go back and look at how much ATP we have produced from a single molecule of glucose. In glycolysis, we're still going to produce 4 ATP through substrate level phosphorylation, but now the 2 NADHs that we produced in glycolysis are the equivalent of each one of these is the equivalent of 3 ATP. In other words, by adding oxidative phosphorylation, we can now say that we have produced even more ATP from glycolysis thanks to the oxidation of NADH and the chemoosmotic gradient that was created by the electron transport chain. The oxidation of pyruvate will also have NADH produced. So when we follow this same train of thoughts of the NADH being an 3 ATP equivalent and the FADH2 being a 2 ATP equivalent, you can see that the amount of ATP that's produced from one glucose molecule is significantly a lot more in the presence of oxygen than when produced by glycolysis alone. That's it for the Chapter 6.2. Thanks to y'all so much for watching. Bye.